All right. Hello, everyone. Today, I want to talk about the future of genetic sequencing. I started my journey with DNA about 7 years ago after my dad was diagnosed with a genetic condition, and I tried to get tested to see if I'd inherited it. Despite being surrounded by literally a plethora of sequencers in the lab I was working at, and having great health insurance at the time, there was simply no way for me to get tested out waiting months for my results and spending north of $8,000. Very frustrated with this, I started to talk to absolutely everyone and anyone I knew in the sequencing space and found that it's actually far from alone. So today's sequencers are these very large, expensive, complex, labor-intensive machines that cost millions of dollars. As a result, all samples have to be shipped to these centralized sequencing cores in order to process. This is very frustrating to researchers who need to deal with the complex sample prep required for next generation sequencing. It makes carrier screening actually impossible for the 99% of genetic testing clinics who can't afford the $1 million sequencer, and even leads to the destruction of up to 30% of agricultural yields annually due to delays in crop pathogen diagnostics. So I kind of assembled a team of researchers out of Northwestern, and they joined me to create the solution, the Acorn Lab. While most sequencers are built to take 10,000 samples and look at one mutation site, our sequencers were built to take one sample and look at 10,000 mutation sites. As a result, we were able to fully automate sample prep, miniaturize the device, and lower the cost of the sequencer from over $1 million to just $5,000. And the key that enabled us to offer these unique and novel features is our patent-pending solid-state nanopore chip. We use a unique chip geometry, the thermoelectric properties of a 2D material, and a bimodal signal processing method to deliver long-read sequencing without compromising on speed or accuracy. Our competitors are focused on creating sequencing solutions for wealthy research institutions who need to do high volumes of targeted testing, making our sequencer the only viable option for clinics, labs, and ag tech groups who need portable and affordable rapid sequencing. While our competitors are developing better and better supercomputers, we developed the smartphone for sequencing. Additionally, introducing a razor and blade business model into the sequencing space has enabled us to offer a lower upfront cost by making the majority of our profit not off of the sequencer, but off of the cartridges and testing and consumables used with every single run. To put the numbers in perspective, we have about 100 sickle cell disease clinics in our network who have signed pre-orders, letters of recs, and letters of intent. If we were to launch to all 100 clinics, we'd only make about $250,000 from our sequencer sales. However, we make $14.6 million in annual recurring revenue. So our upcoming launch with these clinics and labs and research groups I've been mentioning is far from our full vision. It's actually just the beginning. 7 years ago, I set out to get a genetic test for a condition affecting my family, and I found none. In the coming years, we plan to offer direct-to-consumer sequencing, enabling the millions of Americans like me to get affordable, accurate, and actionable preventative healthcare that they need. But our sequencer isn't just a one-time use device. With our ability to detect bacterial DNA as well as viral RNA, the Acorn Lab is poised to run diagnostics for everyday illnesses. The next time you're sick, take a cheek swab and find out the exact strain of strep throat you have, down to its antibiotic resistances, without needing to leave your home in minutes. To date, we've raised about $2 million in dilutive and non-dilutive funding, and we've actually used it to complete our alpha prototype. So we have our very, very first raw product, and I'm actually very proud to say we've sold our very first unit just a week ago. We completed our initial validation studies and are now right at launch. We are raising about a $5 million seed round to cover our full beta launch this upcoming year. My team includes many of the same researchers and engineers that spun out of the GeneSQ lab at Northwestern University with me back in 2021, as well as a few directors from sequencing companies such as PacBio and IonTerra that we've managed to snag. Together, we bring decades of experience in genetics, nanofabrication, and engineering to create the Acorn Lab. Thanks for listening, and I'd love to take questions.
Thank you for the presentation, and thank you for a clear, simple presentation that I actually sort of understand what you're doing. And kind of, I'd like to know a little bit more about the ask in terms of you have some milestones and you're raising some capital. And can you talk a little bit more where that capital is going to go? and what it's going to take you to commercialize it, some
Yeah.
sense of the real opportunity over the next few years. And do you have any strategic partnerships? I don't know what distribution's like for this too.
Yes, absolutely. So I'll break that down into sort of the series. The 5 million will cover our launch. So I kind of mentioned we have about 100 clinics, labs, and research groups who are within our network. And those just 100 clinics, 100 launches, assuming 10 tests per day per clinic, gets us to that 14.6 annual recurring revenue. So the 5 million will go towards first covering that and then potentially expanding to the relevant to like related groups. Right now, we have partnerships with the Foundation for Sickle Cell Disease Research, as well as the Sickle Cell Disease Association of America. Between these, they have about 80 or so clinics. We have like a few like partner clinics within those. And then we're also now forming partnerships with various research groups in the so fungus research groups, as well as algae and probiotics. And within these groups, we have our one partner clinics, and then they expand to like the larger organization. That's our current setup. Our distributors is like, we do have 100 clinics within the network. We have like 4 main people that we talk to who distribute to those 100 clinics. So we've kind of been taking a little bit more of a grassroots approach of developing relationship with individual clinics, like learning more about the overall organization, meeting the overall organization, and then forming partnerships with them. We're planning to kind of continue that distribution channel through our seed round. Towards our Series A, getting like very long-term big vision, I did mention we do want to expand beyond labs and clinics and move into pharmacies and research groups, and then eventually pursue a direct-to-consumer testing service. That's still something we're working on developing, but that's kind of how we're envisioning the next year or so to go.
Hi, Anna. Great presentation. I have a question or more of a confusion in my mind. Why should I use your equipment as opposed to Illumina, which can give me, breadth of result, right? So, in other words, I know you said your segments are different, but if you can, elaborate on what market segments you're going after,
Yeah.
and how does it translate into the revenue you're projecting?
So the core difference between us and Illumina kind of comes from what sequencer would you use for which purpose? So Illumina fundamentally is going to operate in a different market than Acorn. If you're a researcher and you have like 1,000 mice that you want to see how this gene is manifesting in, you want to use an Illumina sequencer because you want to go ahead and quickly sequence all these mice for these known mutations. You want to be able to do high throughput targeted sequencing. You use the Acorn Lab, and when in cases in which you have one sample you want to run and you want to get the full genome. And because our nanopore chip is built specifically for long read sequencing, we actually are able to offer like full sequencing at a much lower cost. Our tests right now are about $50 for like full long read sequences. And the overall cost of the machine is $5,000. An Illumina sequencer would be about $1 million. So it's a higher upfront investment. It's good for some research cases, but in the research cases which you want a long read, low throughput type test, you would use the Acorn Lab.
So in other words, Acorn can do qualitative but not quantitative. Is that fair understanding?
So we provide more information about one sample, but we currently do not offer like multiplexing. That's something we've looked into is potentially, if you're familiar with ONT, they also do some form of long read sequencing, and they have found a way to offer multiple channels. They can't quite get to the full sort of spread that Illumina can, of Illumina being able to run many samples at once. But they have been able to demonstrate like stacking flow cells in a specific method. We've explored that. For now, the benefit is you want one sample really well done, you'd use the Acorn Lab. You want a lot of samples with a targeting mutation site, you'd use Illumina.
Last question. I have a question. Can you give me some sense of what your gross margins are going to be? I know you talked about sort of the razor blade model. So can you give me a sense both on equipment and razor blade gross margin?
Yeah. So So right now we're pricing the Acorn Lab at about $5,000. It costs about $2,500 to develop. So we make about 50% margins. That's one-time cost, not where we're making the majority of our profit. Our cartridges themselves, the whole consumable package for us is about $10, and we're not doing with like bulk orders. We're still doing low volume orders. So I think this will go down significantly. We're able to sell at about $50 per test. So 80% profit margins on the consumables.
Anna, thank you very much.